So in the UF Forest Entomology collection, we have at least 20,000 vials of beetles. And in those vials, there is more than 150,000 beetles, which is the biggest in the world. I study bark and ambrosia beetles. It is important because there are thousands and thousands of species. Most of them are completely harmless and just live in the forest, but there's a few of them which are really big pests and cause huge problems around the world. Sometimes it's quite easy to tell these bark and ambrosia beetles apart, but sometimes you need much more data to figure out which ones are pests, which ones are not, and to kind of understand a bit about their evolutionary history. We have a sequencing core which has a lot of the new fancy machines. The Illumina MySeq, which will give you tens of millions of reads, and it's really useful for studying bark and ambrosia beetles. There's the NovaSeq, which is brand new, the latest Illumina one, and that one can generate thousands of billions of base pairs, hundreds of genomes on each run, which is really exciting. There is also a PacBio machine, which is really good for sequencing long reads at a time. So the sequencing technologies available at UF produce huge amounts of data, millions and millions, even billions of DNA sequences. I really like how in the Forest Entomology Lab, although it might seem like we just study bark and ambrosia beetles, we actually do lots of other stuff. To understand bark and ambrosia beetles, you really have to understand genetics, you really have to understand programming, understand statistics, understand policy, understand forestry, understand trees. You really have to expand your knowledge to understand this one tiny portion of it.